Condensation of cells. The cephalic neural crest is made up of ectomesenchymal condensation of cells. So these ectomesenchymal condensation of cells, they give rise to dental papilla, which in turn forms the dental pulp. Which it forms the dental pulp. Okay, so what is important to remember over here that the cells of the cephalic neural crest, they are made up of both ectodermal and mesenchymal cells. So dental pulp consisting of two type of cells, that is ectodermal and mesodermal cells. Now there are basically four morphologic zones in dental pulp. As we are talking about the structure of the dental pulp, first, if we see the dental pulp under microscope, there are four distinguished morphologic zones that can be appreciated. And the outermost, we would study the zones from outside to inside. The outermost zone of the dental pulp, all of you must be aware, they are made up of cells called odontoblast. So the outermost zone of the dental pulp is called odontoblast layer. So what is actually present? Suppose this is a tooth, any tooth, like this, and this is the pulp organ of the tooth, which is consisting of the pulp chamber and the root canal. This is the entire root canal system that is consisting of this. So this pulp organ, basically, it has an outermost zone of layer of cells called odontoblast layer. This is the outermost zone of the pulp. The first zone, from outside to inside, we would study the pulp. This odontoblast layer, it is a single cell thick zone. So basically, there is a singular file of odontoblasts which outlines the pulp not only in the crown, midroot, but also in the apical one third of the pulp. So the entire pulp organ is outlined by a single layer of cells called the odontoblast layer. These single cell thick layer under microscope, definitely we would be studying the structure of the pulp under microscope. It appears 3 to 5 cell layer thick. What I'm trying to explain, that the pulp in actuality is outlined by a single layer of cells from the crown to the root, but when we study the pulp or see the pulp in morphologic sections of the microscope, what we actually see, the cells appear to be 3 to 5 cell layer thick. This microscopic appearance of the odontoblast layer, it is called pseudo stratified appearance. Or sometimes it is also called palisaded appearance. Pseudo-stratified or palisaded appearance. Why pseudo-stratified? Pseudo means false. It falsely appears to be 3 to 5 cell layer thick, but in actuality there is a single layer of cells that outlines the pulp from the crown to the root. Okay. This is the histologic microscopic appearance of the odontoblast layer. Now, shape of cells, if we talk about, I am discussing the outermost layer of dental pulp, that is the odontoblast layer. The shape of the cells in the crown portion of the pulp, it is tall and columnar in shape. Which cells I am talking about? Odontoblast, the outermost zone. In the midroot area of the dental pulp, they are cuboidal in shape. And finally, in the apical portion of the dental pulp, they are flat and squamous in shape. So the shape of the odontoblast changes from the crown to the root portion, from tall columnar to cuboidal to flat squamous. So there are variations in the microscopic appearance and there are variations in the shape of the odontoblast layer. Now, between adjacent odontoblasts, okay, there are three type of connections. These odontoblast cells, they are not isolated from each other, but they are connected by three type of connections. Means this is one odontoblast, this is another odontoblast. They would be having certain connections between them. So they are not closely opposed to each other, but they are at a microscopic distance from each other, and they have got some mutual connections between them. What are the connections? Three type of connections exist. Otherwise, there are intercellular spaces of about 30 to 40 nanometer wide. So basically, there are three type of connections between the odontoblasts and intercellular spaces 30 to 40 nanometer wide. Okay, these three type of connections which are present between the odontoblast, they are the gap junctions. Okay. These gap junctions are also called nexus. Nexus, nexuses. Then there are tight junctions. And finally, there are desmosomes. These are the three type of connections that exist between the adjacent odontoblast. These gap junctions or nexus, they are made up of proteins called connexin. They are made up of protein called connexin, Connexin, connexin. Tight junctions, they regulate odontoblast permeability. Yes. The permeability of the odontoblast layer is not regulated by the gap junctions, but by the tight junctions. So they are selectively permeable to certain molecules and ions to and fro from one cell to another. So what I am trying to explain, the outermost layer of the dental pulp, that is called the odontoblast layer, which is a single layer of cells outlining the pulp all over. But in the microscope, it appears 3 to 5 cell layer thick. That's why we call it palisaded appearance. And there are three type of connections that exist between them: the gap junctions, the tight junctions, and the desmosomes. And the permeability is regulated by the tight junctions. Okay. Now, what is the function of this odontoblast layer? We all know that odontoblast is the cell that is chiefly responsible for secretion of dentine. So what is the function? The function is secretion of dentine. Dentine is a mineralized tissue which is composed of about 70 to 80% of hydroxyapatite and remaining organic materials. Okay. So basically the secretion of dentine. Now what do they secrete? The odontoblast does not produce dentine as a whole, but it secretes the constituents of dentine that together make up a tissue that is called dentine. So what are the constituents? They secrete type 1 collagen with some traces of type 5 collagen. Mostly type 1 collagen is secreted by odontoblast. They also secrete certain enzymes like acid and alkaline phosphatase. What I am telling you the secretory function of the odontoblast, which results in the formation of dentine. It secretes phosphorin. It also secretes dentine siloprotein and a special compound called nestin. So these are the substances which are secreted by the outermost layer of dental pulp, that is the odontoblast layer. This nestin, it is considered to be the histological marker of dentine. See, basically, dentine is one of the calcified tissue. The other calcified tissues present in the oral region are enamel, cementum, and bone. So there are principally four types of calcified tissues: enamel, dentine, cementum, and bone. Now, how do we know that that particular tissue is dentine? By the presence or absence of nestin. If nestin is identified in a particular calcified tissue, it would be dentine. So nestin is considered to be the histological marker for the presence of dentine, the most exclusive component that is present only in dentine. If suppose nestin is not there in the option, the second best histological marker is phosphorin for the presence of dentine. So this is the outermost layer of the dental pulp or the odontoblast layer, its features and functions. Then the second layer from outside to inside is called the cell-free zone, which is also called cell-free zone of V. Okay. It is basically a 30 to 40 micron wide zone below the odontoblast layer. As the name indicates, cell-free zone, no cellular structures are present in this zone, but it consists of certain capillaries, small capillaries, dendritic cells, certain dendritic cells, and sorry, not dendritic cells, but the dendritic processes. No cells are present there, and it consists of nerve fibers, mostly non-myelinated nerve fibers. They are present in the outermost layer of dentine. That is called cell-free zone of V. The third zone from outside to inside, that is called the cell-rich zone. Okay. What I am telling you, I am telling the morphological organization of dental pulp. It is divided into four zones histologically. The outermost layer is the odontoblast layer, then the cell-free zone, and then the cell-rich zone. Cell-rich zone is also called zone of Hole, HOHL. As the name indicates, cell-rich zone. It consists of numerous cells. Numerous pulpal cells are present over there. What are the different cellular populations present? The most predominant ones, they are the fibroblast. They are the most abundant cells of the dental pulp, always. Okay. And what is the function of fibroblast? All of you must be knowing, aware of it, that fibroblast is the cell that secretes collagen secretion. What type of collagen is secreted by fibroblast? Both type 1 and type 3 collagen is secreted by fibroblast. So if I talk about odontoblast, it is secreting principally type 1 collagen with traces of type 5 collagen. And if I'm talking about fibroblast, it is secreting type 1 and type 3 collagens, which form up the organic component of dentine. Okay. Then the second cells, they are the undifferentiated mesenchymal stem cells. UDMSC, undifferentiated mesenchymal stem cells. They are present in the cell-rich zone most abundantly, but distributed throughout the pulp. Okay, they are not restricted to the cell-rich zone only. They are distributed throughout the dental pulp, but they are most abundant where? In the cell-rich zone. As the name indicates, cell-rich zone, they would be having the highest population of all the available cells in the dental pulp. These are basically multipotent cells, multipotent postnatal stem cells, mostly present in perivascular areas of cell-rich zone. Mostly in the perivascular areas of cell-rich zone. Perivascular means around the blood vessels because they require a rich supply of nutrients for the growth and differentiation of stem cells. The details of the stem cells I am not taking right now. What are the different types of stem cells present? What is the function, etc. That is the subject of another topic. We would see it in some other session. Right now, you just remember that there are multipotent postnatal stem cells present in the dental pulp, which are mostly distributed in the perivascular areas of the cell-rich zone. The third population of cells are the macrophages. Macrophages, they are derived from blood monocytes. Okay. Monocytes are the progenitor cells which are present in the circulating blood. They give rise to macrophages. The macrophages, they have got two functions. As the name indicates, macrophages, one is the primary function and another is the secondary function of these macrophages. The primary function is definitely phagocytosis. Okay. They recognize an antigen or any other foreign material, phagocytose it, and present it to the defense systems of the cells. Phagocytosis. The secondary is immune reaction. Immune reaction. These macrophages also contribute to the innate immunity of the dental pulp by way of major histocompatibility complex class 2 molecules. These molecules are present on the surface of these macrophages that contribute to its secondary immune function. What is the primary function? The primary function, as the name indicates, is phagocytosis of the foreign materials, other toxins, etc. Another cellular elements which are predominantly present in the cell-rich zone, they are the dendritic cells. These are also called Langerhans cells or simply APC, that is antigen presenting cells. We are discussing the cellular components of dental pulp. So whenever the pulp is healthy, a normal uninflamed pulp is present, these antigen presenting cells, they are distributed mostly in the periphery of the dental pulp, outer zones, the periphery. In the case of inflammation or infection of dental pulp, there is some inflammation or infection of the dental pulp. These cells, they migrate to the center of the dental pulp. It is very logical also because these are the principal immune cells. What are these? They are the principal immune cells. So whenever there is some invasion of the dental pulp by way of microbes from caries, etc., definitely the outer zone of the pulp will be affected first. So these antigen presenting cells, they recognize the antigens, take them to the defense system. In the case of inflammation, migrate to the center. Another cellular components of the dental pulp, they are the lymphocytes. Lymphocytes are what? They are the white blood cells. All of you know that there are two types of lymphocytes present in the body, the B and the T lymphocytes. There is no conclusive evidence of the presence of B lymphocytes, but T lymphocytes are definitely present in the dental pulp. Okay, what are the T lymphocytes? T8 suppressor cells. They are the predominant T lymphocytes present in the dental pulp, and as the name indicates, T lymphocytes, they contribute to what type of immunity? Cellular immunity of dental pulp. Okay. And finally, The cells that are present in dental pulp in certain special conditions, they are the mast cells. The mast cells are absent in normal healthy pulp, but they are present in cases of inflamed pulp. Okay, we are discussing the third zone from outside to inside, that is the cell-rich zone. As the name indicates, cell-rich zone, various cellular components are present in the dental pulp. These are the six predominant type of cells present in the dental pulp. The mast cells, they consist of granules. Okay. These granules of the mast cell, they contain two substances, H and H. You can remember like that. It's a mnemonic. Histamine. Okay. What is histamine? Is a vasodilator. And heparin. What is this? It is an anticoagulant. So whenever there is inflammation of the pulp, some of you must have seen that while doing root canal treatment of an inflamed pulp, there is copious bleeding from the pulp. That is unstoppable even after application of copious amounts of sodium hypochlorite. It is a clinical observation. An inflamed pulp bleeds copiously. At times, it is so copious that it feels like we have entered an artery. Why it happens? Because of the presence of mast cells in inflamed pulp that releases heparin and histamine. Vasodilation is there, so there is stagnation of blood. More blood is pooled in the pulp, and heparin is there that causes the blood to flow continuously for a longer time without being coagulated. So it is clinically correlated to the presence of mast cells in the inflamed pulp. These are the cellular elements of dental pulp. Now, the innermost zone of the dental pulp is the pulp proper. Pulp proper. This is the innermost zone of dental pulp. As the name indicates, pulp proper, it is basically histologically a loose connective tissue. Histologically, it is a loose connective tissue. So, what type of tissue is dental pulp? The answer would be it is a loose connective tissue. As the name indicates, pulp proper, it consists of cells. What are the different cells? We have already seen, but the highest concentration of these cells would be present in the cell-rich zone. Then it has got large blood vessels and nerve fibers. These are the structures, components of dental pulp into four morphologic zones and their important characteristics. Now, we have been seeing that numerous nerve fibers are present in the dental pulp. Mostly, what type of fibers are present, how do they act, we would see subsequently. So, after seeing what is the structural organization, we would talk about the innervation of dental pulp. Innervation of dental pulp. Now, dental pulp is basically innervated. The pulp organs, whether they are maxillary or the mandibular, primary or the permanent teeth, they are all supplied by the trigeminal nerve. Okay, trigeminal nerve is basically the fifth cranial nerve. That is a mixed nerve. It consists of both sensory and motor fibers. Trigeminal nerve. So innervation basically starts from the superior cervical ganglion. Superior cervical ganglion, which is present in the middle cranial fossa, petrous part of the temporal bone. Okay, superior cervical ganglion, the ganglion of the trigeminal nerve. Okay, so the sensory innervation, of this trigeminal nerve to the pulp. The sensory innervation consists of two type of fibers, the A fibers and the C fibers. Okay, I'm talking about the innervation of dental pulp, which is derived from the trigeminal nerve. The sensory part of this innervation consists of two type of fibers, A and the C fibers. The A fibers, they are the myelinated fibers, and C fibers, they do not have a myelin sheath, they are non-myelinated. And from the basics of the physiology, you must be knowing that myelinated nerve fibers, they are fast conducting, and C fibers, which are non-myelinated, they are slow conducting. These are the two type of sensory fibers which are present. Now, if we look at the further details of these sensory fibers, the A fibers, which are present in the pulp, can be subdivided into two categories. Okay, they are the A beta fibers and the A delta fibers. These A fibers, which are the myelinated fibers, together constitute about 10% sensory fibers. They make up about 10% of the sensory innervation of dental pulp. Now we are talking about sensory fibers, means that carry certain sensations from the pulp to the brain so that the person perceives it and there is according reaction to that stimulus. A beta fibers, they constitute just 10% of the A fibers, and majority, that is 90% of the A fibers, are A delta fibers. The sensation of proprioception or pressure reception is carried by the A beta fibers. On the other hand, the A delta fibers, they carry the sensation of pain, temperature, and touch. Okay, pain. This is the principal sensation that is carried by the A delta fibers, and we know that majority of the A fibers are A delta fibers. These A fibers, they are present in pulp periphery. In the periphery of the pulp, near dentino-enamel junction. Okay, in the outside of the pulp. Okay, up to the, sometimes extending up to the dentino-enamel junction from the pulp. Periphery of the pulp, these are present. Okay, these proprioception or the pressure reception is caused by the A beta fibers, and we have seen that A beta fibers are very less in the present in dental pulp. So it causes poor localization of pulpal pain. The question is asking you why it is difficult for the pain of pulpitis to be localized. Pulpitis, not periodontitis. Okay, pulpitis. There is inflammation of the pulp, whether acute or chronic. At times, it is difficult to localize only particular region of the tooth pain. The mouth is sensitive, but the patient is not able to localize that this particular tooth is offending, causing me pain. Why? Because there are very less proprioceptors in the dental pulp. A beta fibers are very less in number, so the pain of pulpal origin is difficult to localize. Once the treatment is not given in the initial stages of pulpitis, it goes down, causes pulp degeneration, and then apical periodontitis. So whenever there is inflammation of the PDL or periodontitis, slight touch or percussion elicits pain to the patient. That's why we call it situation called tenderness on percussion. So tenderness on percussion basically indicates periodontitis, apical periodontitis, and not pulpitis. Okay. So When there is initial inflammation of the pulp, the pain of pulpal origin is difficult to localize because it consists of very less A beta nerve fibers. What type of pain is carried by A delta fibers? It is a short and sharp pain. The quality of pain that you may have experienced at times when you bite upon a very cold stuff like ice cream, you feel a momentary sting, a pain sensation for a couple of seconds. That sensation is carried by the A delta fibers, which are present in periphery of the pulp and can extend up to the dentinoenamel junction. The C fibers, on the other hand, they carry the sensation of only and only pain. Okay, and 80% of the fibers present in the dental pulp are C fibers. Okay, only 10 to 20% are A fibers. 80% of the fibers are non-myelinated C fibers that carry the sensation of pain only. So if the question is asking you which is the principal sensation carried by dental pulp, it would be pain, because both A delta and C fibers, they carry the sensation of pain. That's why these C fibers, they are also called principal nociceptors. Nociceptors means those carry the sensation of pain. Proprioceptors, those carry the sensation of pressure. Now these C fibers, they are stimulated by acidic pH of less than 6, temperature of greater than 110 degrees Fahrenheit, and certain chemicals like capsaicin. What? C fibers which carry the sensation of pain, they are stimulated by pH less than 6, temperature 110 Fahrenheit and above, and chemicals like capsaicin. So there are numerous methods by which the C fibers can be stimulated, and all sorts of stimulation will result in which sensation? Sensation of pain. So another name for the pulpal C fibers is polymodal nociceptors. Why polymodal nociceptors? Because there are different modes, numerous modes of stimulation of these C fibers. This is about the sensory innervation of dental pulp, and the quality of pain that is carried by C fibers is dull and throbbing pain. The quality of pain is dull throbbing pain. Okay, pulsatile type of pain that is less bearable. It is less bearable pain for the patient as compared to the short, sharp pain caused by A delta fibers. So these are the different type of sensory fibers present in the dental pulp, their location, their constitution, and the functions and the type of sensations that they carry. Now, pulp also consists of, apart from sensory fibers, they consist of postganglionic, sympathetic nerve fibers, postganglionic sympathetic nerve fibers are present in dental pulp. So, if the question is asking you about the innervation of pulp, it would be both sensory fibers, two type of sensory fibers we have seen, and it would be postganglionic sympathetic nerve fibers. The presence of parasympathetic nerve fibers in the dental pulp is not documented. There is no substantial evidence to say that there are parasympathetic nerve fibers present. It is only the postganglionic sympathetic nerve fibers, and they have the function of neuromodulation. See, basically, anywhere in the body, if we talk about innervation, has got two functions. One is sensory, means from the periphery toward the CNS, and another is motor. That is, the brain gives signal and we move our limbs, etc., do activity. Similar thing is happening in the dental pulp. There is a sensation carried by the A delta and the C fibers and the A beta fibers, and there is signal coming down through the postganglionic sympathetic nerve fibers that have the function of neuromodulation. There is no motor function in the pulp. The pulp does not move, but through postganglionic sympathetic nerve fibers, there is neuromodulation, and this happens by secretion of certain chemicals by these nerve fibers called neuropeptides. As the name indicates, nerve proteins. Secretion of neuropeptides. The principal neuropeptides present in dental pulp are CGRP and substance P. These are the principal neuropeptides of the dental pulp, mostly secreted by sensory nerve fibers. CGRP has the full form of calcitonin gene related peptide. So instead of the motor function, the pulpal nerve fibers has the function of neuromodulation by way of these neuropeptides. These are the sensory innervations and the efferent innervations of the dental pulp. Now we talk about blood circulation of the pulp. Now, the blood circulation of the blood pulp is derived from the maxillary artery. Both the maxillary and the mandibular teeth, they are supplied by the maxillary artery. This maxillary artery is a branch of external carotid artery. It is a branch of what? External carotid artery. So dental pulp has a copious circulation, so the resting rate of blood flow. If we have to talk about the resting rate of blood flow in the dental pulp, it is in the range of 15 to 60 ml per minute per 100 gram tissue, per 100 gram of the dental pulp. Important thing to remember over there is I am giving you the figure for 100 grams of the dental pulp. If the question is asking you per gram of the dental pulp, divide these figure by 100, you get 0.15 to zero point 0.6 ml per minute resting rate of blood flow. Now, whenever there is blood circulation, blood consists of hemoglobin that carries the oxygen. So, another way is rate of oxygen consumption of dental pulp. What is the rate of oxygen consumption of the dental pulp? It is in the range of 3.2 ml plus minus 0.02 ml per minute per 100 gram tissue. Both the figures I have given you for 100 gram of the tissue. Okay, the rate of oxygen consumption. So more the blood flow, more would be the oxygen consumption, greater would be the metabolic activity, and the resting rate of blood flow to the dental pulp is one of the highest amongst the tissues present in the body. That's why the pulp, whenever there is inflammation of the pulp, there would be increased blood flow to the pulp, isn't it? And pulp is encased in dentine and enamel that are hard encasements. So pulp has got no expansion and it causes severe pain of irreversible pulpitis to the patient. That's why the dental pain is so unbearable. Okay? So these are the rate of blood flow and the resting rate of oxygen consumption of the pulp. Now there are certain materials like dental amalgam. Okay. Materials like dental amalgam. calcium hydroxide, zinc oxide, eugenol. They all reduce the oxygen consumption of pulp. Means they basically reduce the metabolic activity of the dental pulp. Similar is the orthodontic force. Okay. Presence of braces, orthodontic force also reduces the metabolic activity or rate of oxygen consumption of dental pulp. Now, whenever there is circulation in the pulp, it would result in pulpal pressure. So, a normal pulp or a healthy pulp has an intrapulpal pressure of about 10.2 millimeters of mercury column. Definitely, there is circulation, there's blood circulation in our body. We have got our normal blood pressure. Similarly, whenever there is circulation in the normal pulp, the pulpal pressure is 10.2 millimeters of mercury. If we are measuring the pulpal pressure by the water column and not by the mercury column, it would be 14 centimeters of water column. Okay, normal. So, what happens in the inflammation of the pulp? We know that whenever there is inflammation in the body, there is development of classical signs like rubor, pallor, delor, etc. Isn't it? Similar thing happens whenever there is inflammation of the pulp. Two important changes happen in the pulp physiology. One is increased vascular due to liberation of chemical mediators of inflammation, and there is vasodilation. The blood vessels dilate and the vascular permeability is increased. These two phenomena together contribute to increased intrapulpal pressure. So, whenever there is inflammation of the pulp, the intrapulpal pressure would rise. And since pulp is confined in a low compliance environment, low compliance means there would be no volumetric change in the pulp canal system. That increased blood volume, that increased vascular permeability causes immense pressure, and the patient feels throbbing, intractable pain in cases of irreversible pulpitis. Intrapulpal pressure rises to nearly 30 to 35 millimeters of mercury in cases of pulpal inflammation. Okay, so what is the normal intrapulpal pressure? I have told you. And what is the pulpal pressure in cases of inflammation? I have told you. And what is the reason for increased intrapulpal pressure? These two are the reasons. Okay, previously one concept was there, pulp strangulation. Okay, third reason previously proposed was pulp strangulation. Strangulation means to cut off, isn't it? They proposed that whenever there is increased intrapulpal pressure, the pulp becomes strangulated and it dies. But the modern concepts do not favor pulp strangulation theory. Only two factors or two phenomena that happen during inflammation of the pulp are increased vascular permeability and vasodilation that contribute to increased intravalvular pressure at types of pulpal inflammation. These are the different aspects of the vascularity of pulp, how it is supplied, and what are the changes that happen in the cases of this. Now, there is a clinical observation called phantom tooth pain. Phantom tooth pain. This is due to a phenomena called deafferentation. Efferent means sensory, efferent means motor, isn't it? Deafferentation. Phantom tooth pain is basically defined as persistent pain in RCT treated tooth, a clinical phenomena of persistent pain in a root canal treated tooth. Persistent pain, any pain greater than equal to 6 months duration is defined as persistent pain. RCT treated tooth means all the nerve fibers have been removed from the pulp. It is deafferentation has been done. But still, there is pain in a patient. RCT treated tooth means there are no procedural errors. A well treated tooth is there, and in such situation, patient complaints of pain is known as phantom tooth pain due to phenomena called deafferentation. It has an incidence of 3 to 6%. That is quite high. And what is the reason for such is increased expression of C-phos G on pulpal nerve fibers. Especially in cases of symptomatic irreversible pulpitis, patient is having spontaneous throbbing pain, and we have done root canal in such a tooth, then there is a 3 to 6% incidence likelihood of persistent pain after root canal. A good root canal that is adequate in all aspects. Patient complaints of pain is known as persistent pain, and the phenomena is phantom tooth pain. Phantom tooth means no nerve fibers are present, but still the pain, spontaneous pain is there to the patient. Okay. And this is due to increased expression of C-phos gene. Further details are not required for you. You just remember what is the reason. There's increased expression. And how can we manage them? What is the treatment? There is no specific treatment for this. Only long-term analgesics can be given to the patient. Okay. These are the different aspects of your pulpal structures, starting from the zones, development of the pulp to the four morphologic zones, the different constituents and functions. Then we have talked about the innervation, and then we have talked about this blood vessels and its related phenomena. Now, as the person advances, age advances, there are certain pulp calcifications that happen. These pulp calcifications, they have incidence of nearly 50% into the population. These calcifications, they can be diffuse calcifications, or they can be in the form of pulp stones, concentric. Concentric pulp stones, diffuse calcifications. Okay, if this is the pulp organ, there can be development of calcifications in the concentric forms. Okay, diffuse, they are called false, and concentric pulp stones, they are the true pulp stones. They have a lamellated structure. Lamellated structure means it is organized into layers or lamellae. At times, it can also consist of dentinal tubules. Tubular structures are present. Okay. They are more common in the coronal pulp, and diffuse calcifications are more common in the radicular or the root portion of the pulp. The clinical significance is that during root canal therapy, we have to remove these pulp stones for better access to the root canals, because they interfere with the placement of instruments into the root canal properly. They are the metamorphic calcifications of the dental pulp, age changes in the dental. Another thing happens due to age. With increased age, the cells decrease in number. We have seen the different cells of the dental pulp, starting from fibroblasts to the mast cells. All of them decrease in number. However, there is increased number and size of collagen fibers. Type 1 collagen fibers, but traces of type 3 collagen fibers are present in the dental pulp. So it consists of both type 1 and type 3 fibers, and their size as well as the number increases with age, and the cellular components decrease in size with age. So these are the different aspects of the structure and functions of dental pulp. That was the topic for today. Okay. The trend in these days MDS has been, any MDS examinations have been sticking more to the basics. You have to be clear with the basic of each and every subject. And that is the basic focus of our 360-degree program. We would be taking one topic at a time so that a more comprehensive preparation is done. And it is my humble suggestion that as and when the topic is covered, you please open the MCQ books and start doing the relevant MCQs so that it is strengthened in your memory, the concepts. And I have received a message that a couple of students joined a little late. For them, I am just revising the beginning of the session for 1 or 2 minutes. So what I started today, I started with the structure and functions. Structure and function of dental pulp. And we talked about the embryologic origin of the dental pulp. It is from the cephalic neural crest. Okay. And the neural crest is made up of ectomesenchymal condensation of cells, and it has got two parts, the cephalic part and the caudal part. The dental pulp is derived from the cephalic part of the neural crest. The cells of these neural crest are ectomesenchymal cells, so both ectodermal and mesenchymal cells are present in the dental papilla that give rise to the dental pulp subsequently. If we talk about the structural organization of dental pulp, it is basically divided into 4 zones. Morphologic zones means that can be identified under histologic sections. The outermost zone of the pulp, it is called the odontoblast layer. Okay. It is the odontoblast layer. This odontoblast layer, as is shown in this diagram, okay, it is a single layer of cells that outlines the pulp from the crown to the root. But whenever we see this odontoblast layer under the microscope, it appears 3 to 5 cell layer thick. And this microscopic appearance of the odontoblast layer, it is called pseudo-stratified appearance of odontoblast layer. Pseudo means false. So falsely it appears to be 3 to 5 cell layer thick. This appearance is also called palisaded appearance of the odontoblast layer. Now these odontoblasts that outline the pulp, if we talk about the morphologic shape, in the crown portion of the dental pulp, they are tall and columnar in shape. In the midroot portion of the dental pulp, they are cuboidal in shape. And finally, in the apical portion of the dental pulp, they are flat and squamous in shape. So the shape of the odontoblast, that is the outermost layer, changes from the crown to the midroot to the apical 1/3. And this adjacent odontoblast consists of 3 types of connections between them. Okay, these are the odontoblasts, some 30 to 40 nanometer wide. This is nanometer. Okay, intercellular spaces present between them. The 3 types of connections, they are the gap junctions, the tight junctions, and the desmosomes. Gap junctions, they are also called nexus, and they are made up of a protein called connexin. The tight junctions and desmosomes are the other 2 types of connections. The tight junctions are important in the sense they regulate the permeability of the odontoblast layer. There is selective passage of molecules and ions from one odontoblast to another, from odontoblast layer to the subodontoblastic layer, and this passage of molecules and ions is regulated by the tight junctions. I think this was all that was left for the students that joined late. And with this, I'm concluding to this session. I hope it has been comprehensive enough to understand and introduce the subject to you. My best wishes to you all. Good night. Happy reading. Thank you very much for being connected.